Good evening, everyone. So we'll wait for 2 more minutes, then I'll start the session. Is my voice audible to everyone? You can just reply yes or no in the chat box. Okay, thank you. So good evening, everyone. I welcome you all to this week 3 of problem solving session for the NPTEL course Cell Culture Technologies. And in between, if I'm discussing any question, if you have any doubt, or if you know the answer to that question, you can just write it in the chat box. Don't unmute yourself and speak because then it will create a lot of disturbance. Also, if you have any other questions that you would like to discuss, you can put those questions in the chat box. And once I stop discussing the questions that I have, after that I can take up those questions and answer them. So let us begin. The questions that I'll be taking here, the 10 questions, are from a model assignment for the same course. And this the type of questions that I've taken will help you in solving the week 3 assignment of the current cell culture technologies course. So let's begin with the first question. What is 20 in a T20 flask? So there are four options: length of the flask top, height of the flask side, area of flask base, and last option is volume of the entire flask. So if anyone knows the answer, they can just type it in the chat box, and then I'll discuss the answer in detail. Okay. So here the correct answer is, in a T20 flask, this 20 or any number that will be here for that matter will denote the area of the flask base. So in cell culture, what happens is we use different kinds of vessels or culturing dishes or flasks in order to cultivate our cells. So these flasks which you are seeing here are actually called tea flasks. Now, as you can see here, there are different sizes of tea flasks which are present. One is a very small one is intermediate, and one is a big flask. So here, these flasks are denoted as T20, T25, T75, T125, and so on. So here the numbers which are written here after the T 20, 25, 75 or 125, they represent the area of your flask base where, like basically this area where your cells will attach and they will grow. Now tea flasks are the most commonly used flasks used for culturing cells. However, there are other dishes also which are used for growing and maintaining cells, and they serve a slightly different kind of purpose. For example, these round dishes that you are seeing, these are petri dishes. And these small plates you are seeing with wells. So this is a classic 96 well plate. This is a 6 well plate. This is a 12 well plate, and this will be a 24 well plate. Now the most important question is, why do we use different kinds of these vessel systems or different kinds of culturing dishes or plates or flasks for growing cells? So the reason is, these tea flasks are usually used for maintaining cells because, as you can see, they have these caps here and this neck here. So they help in preventing contamination. Whereas if you compare contamination in a tea flask to that of a petri dish, which is something like this. So here these petri dishes are contamination prone because you'll open the lid and you'll keep using them. However, for ease of passaging cells, for ease of transfection and other things like that, these petri dishes are favored over tea flasks. So tea flasks are usually used when we have to maintain and culture cells for a long passage of time. And then there are different kinds of plate which you can see here, which are used in the laboratory. There's 96 well plate, 6 well plate, 12 well plate, and 24 well plate. Now these are used according to the kind of assay that you are going to perform. Suppose if you have some infectivity assay for which you do not need to culture the cell in such a big amount, because here what will happen if you perform that assay in such a big like big plate or big flask, then you will be wasting your cells, you will be wasting your regions, you will be wasting a lot of media. So for that, depending on the kind of assay that we are going to put, we determine what kind of like plate or what kind of culture dish or what kind of vessel we'll be going to use. Now, another important, when it comes to tea flasks, as I said earlier, tea flasks are uh, used for maintaining cell lines, like or for culturing or subpassaging cell lines. So for that, as I showed in the previous problem solving session, we talked about something which is called confluency. So I hope you remember what confluency is. You can again look at this panel of figures. So here, as you can see, the confluency is somewhere around 10%. And if you compare this panel on the left to this panel on the right, where it is written that confluency is around 30%, then you can clearly see the difference. Then here, look how the cells are looking when the confluency has reached 50%. 50% meaning it has covered half the area of the culture vessel or the culture dish in which your cells are growing. And then this is something which is where confluency is 90%. So what happens is, once you plate your cells, suppose you plated your cells at this day 0, then what will happen? There will be a initial small lag phase of growth where you'll find that there's no growth happening or the growth is happening very slowly. And once your cells adhere to the surface and once they get the proper nutrients and start growing, you'll see a gradual increase in confluency. Like first it will reach 10%, then 30%, then 50, and then 90%. So here what happens is there is a log phase of growth where you will see an increase in confluency. Now, once your cells are in this log phase of growth and they are somewhere in this 85 to 90% confluency range, it is always the like best way to subculture them, because once your cells reach 100% confluency, then it will start reaching a plateau. And here what will happen is, if your cells have reached 100% confluency means they have covered the entire surface area of your culture flask. Then what will happen? Then there will be no area left for new cells to divide and grow. And then if you leave your plate at such a kind of condition, then your cells will start dying or slouching off. So the best point at which you can subculture is when your cells are in 85 to 90% confluency state. Now, uh, so this is how when we are maintaining cells in T flask. So this is the basic like diagram for when your cells are growing from your day 0 till the time it takes to reach 85 to 90% confluency, and at what point you should subculture them or passage them. So the next question is, based on question 1, the dimension 20 in T20 is it meter, is it millimeter, is it centimeter square, or is it cubic diameter? So if you know the answer, you can put it in the chat box. So as I mentioned earlier, the if you look at the answer to the first question, the answer is this 20 in T20 flask is the area of the flask base. So if it is area, then what will it be? Yes, so the correct answer is it will be centimeter square. So this 20 is 20 centimeter square area. As you can see here, there's a hint in the question itself. Like the unit of area will be square, so it is centimeter square. As I mentioned earlier, different kinds of tea flowers have different kinds of area, and depending on the kind of culture or the amount of cells or culture we need for our assay or for maintenance, we can choose from the from these different kinds of flasks which have different area. So here, as you can see, the first flask, the top flask is 25 centimeter square. The second is 75 centimeter square, and so on. And how these flasks, what will be the name of the flask here? This will be T25. This will be T75. T175 and T225. So it is not just that the tea flasks have different kind of different areas. The different other kind of dishes that I discussed, they also have a different area and they serve different purpose. For example, if you look at the table here, if we just consider about a tea flask, a T25 flask has an effective growth area of 25 centimeters square, and the cell culture medium that, like the amount, the volume of cell culture medium that can be used in a T25 flask will be 5 ml. But if you go higher, if you go to a T75 flask, which has an effective growth area of 75 centimeters square, then what will happen is you can use more media and you can grow more cells, because here the volume will be up to 15 ml. Similarly, if you take a T150 flask, then you can go up to 30 ml. The same is with dishes. By dishes, I mean those circular plates. So as you saw in the previous diagram, the dishes are also different. Like they are of different sizes depending on their effective growth area, and depending on that, the different volume of cell culture medium which will be required will be different. Same goes for 6 well plate, 12 well plate, 96 well plate. Now here, since we are discussing about tea flask and like since we have talked about passaging or subculturing of cells, I have included a video where I'm going to show you how subculturing takes place in a tea flask. So this what you are seeing here, this is a cell culture hood. I discussed about cell culture hood in my previous lecture, so you can take a look at that. And here, as you can see, so in the previous assignment, we talked about the basic flow of the cell culture hood. Like in this area, you should have your clean or autoclaved equipment. Like on the extreme left side, then in the middle, you should have your working equipment, because this will be your working area. This is the place where your hands are going to be, and on the right side will be your discard. So here in this picture, this this bottle that you are seeing with this pink liquid is the discard, and here you can see this is a tea flask. And now we can watch the video and see how subculturing takes place in a tea flask. So this is a stere pipette. As you can see from, like, in order to take out the old media, this cap was opened, and here, since our cells are growing like at the surface, in order to remove any spent media, or any leftover media, what do is we give a PBS wash. Now PBS is phosphate buffered saline, and it maintains the osmolarity of the cells. And once you have given the PBS wash, as you saw from this bottle of trypsin, we added trypsin so that the cells becomes detached. And in this condition, if you look under the microscope, the cells will appear round, as you can see here, because they have become detached. Their ECM has become detached from the surface due to action of trypsin, which is an enzyme. Now, once your cells have become detached or rounded, what we should do is we should add the media. So this media has serum. or FBS, which is fetal bovine serum, and it inhibits the activity of trypsin. Hence, what will happen will be like whatever trypsin is remaining in your dish or your vessel, it will get inactivated. Now, after that, since your cells have become detached and they have come up to the media, what she has done is she has taken it out in another tube, which is a falcon tube. First, she has taken it in the falcon, and she's removing whatever extra media or extra culture is there. She's adding more media and pipetting it up and down, as you can see. Now she has taken some amount, and this is something which is called a hemocytometer. We'll discuss about this in detail in future classes. So a hemocytometer is used to count cells. Here you can, as you can see here, there were some dead cells and some live cells. And she was using a special type of stain, which is the trypan blue stain. What trypan blue does is it gets inside dead cells because they are dead, and it will not be able to stain the live cells. So you can look under the microscope and you can count your live versus dead cells. And once you have a count, you can plate your cells. Suppose you want to plate 1 million cells in your T20 flask. So you can plate your cells because you will have that count now. Once you have counted that cell, counted the number of your cell in suppose 1 ml, then accordingly you can scale that up, that if 1 ml has this number of cell, then like 5, what will be the number of cell in 5 ml, and accordingly you can plate. So once you have plated the cells again, what the next step is to keep the flask which has your new detached cells in an incubator which can grow at 37 degrees Celsius, and appropriate level of CO2, which will be 5 to 10% CO2. So this is how a subculturing in a basic tea flask looks like. I've shown all of you this video so that you can appreciate how in an actual laboratory this process takes place rather than just reading about it or hearing about it. So moving on to the next question. The next question is, to ensure carbon dioxide exchange in a T20 flask, what do you have to do? So we discussed this in the previous lecture. If you have missed this, if you have missed those lectures, you can go and visit those again in the YouTube channel. So what happens is CO2 is a very important gas in maintaining the pH of cells or cell media. So we have an entire bicarbonate buffering system where CO2 plays a very important part, which we have already discussed in the previous lecture. And because it has to maintain the physiological pH, which is around 7.2 to 7.4, and if the pH is not something around this, then your cells will have problem growing, or they may not grow at all. So in your T20 flask, as you saw in the previous diagrams, so your T20 has a lid here, right? So what do you do with this lid? Because you have to ensure proper CO2 exchange and proper gaseous exchange, not just CO2 but oxygen as well. So what will you do with this lid? Will you close it tightly while in incubator? Will you leave it slightly open while it is in the incubator? Or leave it fully open in incubator, or none of the above? Someone is saying my voice is not clear. Is it the same for everyone, or is it fine for everyone? Okay, it's clear. Uh, okay. So, yeah, I can see the answers. The correct answer is B. What you will do, you will leave it slightly open in the incubator. You will not close it very tightly because then there will be no gaseous exchange. You cannot leave it fully open because then it will be prone to contamination. You have to leave it slightly open while it is in the incubator. So in the T20 or the tea flask, we have these different kinds of vented lids. Vented lids meaning, as you can see, there are some holes in the lid, and here we have a kind of filter membrane which can be 0.22 micron, or like depending on the manufacturer, the filter membrane size can vary. So what these vented lids do, like even if you close your lid tightly, then because they have these filter membrane which allows the gaseous exchange, and they will filter out any kind of contamination. Suppose it is if it is 0.22 micron filter, then it can filter out bacteria and things like that. And here, so this is to ensure proper gaseous exchange, proper CO2 exchange. Here in this flask also, you can see there are some holes, and uh, and behind, if you flip the lid around and see, you will see this filter membrane. So as you can see in the tea flask here, the gaseous exchange is taking place through the vented cap. But you have to make sure that even if the cap is vented, you cannot close the lid very tightly so as to stop any kind of gas gaseous exchange. You still have to leave it slightly open. So moving on to the next question. The next question is, matrigel coating is done prior to cell plating. What is matrigel? So now again there are four options. Is matrigel a cleaning agent? Is it an ECM and extracellular matrix, antibacterial coating, or cell suspension? Yes, so the matrigel is an extracellular matrix. So if we talk about how matrigel looks like when we are going to use it in a laboratory, so this is what it's going to look like. So this matrigel has phenol red, that is why you can see this red color. And it is always important to like keep the matrigel at -80 or in ice at 0 degree or 4 degrees Celsius. And that is because if you increase the temperature, the matrigel starts forming a matrix. So that is why, as you can see here, it is kept in ice. And then you can alicot these matrigels to prevent contamination or to prevent degradation. So what is a matrigel? So a matrigel is a matrix. It is an extracellular matrix, and it is a solubilized basement membrane, and it is extracted from a kind of mouse sarcoma. So this is the specific term. It is an EHS mouse sarcoma, and it has very rich ECM protein. So as we discussed in the first lecture assignment or in the first lecture videos, the ECM is highly complex as it has different kind of proteins, it has different kind of glycoproteins and collagen. So this matrigel also has different kind of ECM proteins such as laminin, collagen 4, heparan sulfate, proteoglycans, intactin, and then a different number of growth factors. So what happens is your because this your matrigel has different kind of ECM proteins, and it is highly rich in growth factors. what will happen is if you coat your culture dish, if you coat your culture dish with matrigel, then you will see your cells growing similar to how the cells grow inside the body. For example, matrigel is extensively used for culturing epithelial cells, and in the body, when epithelial cells grow, they form these like tube like structures which are like the web of a spider, as you can see here in the S panel. And if you are culturing epithelial cells outside, not in matrigel matrix, suppose in some other kind of matrix, then what will happen? You, your cells will adhere, they will grow, but they will not. You will not see them forming these special kind of tubules or special kind of network that you see with matrigel. And this is how the cells grow inside the body. So matrigel can mimic the exact kind of environment which the cells get uh, to as they grow inside the body, which is not true for like any other synthetic matrix. Now again, just so you can appreciate how the matrigel coating is done, because I'm constantly saying that we do matrigel coating on cultured dishes for your cells to grow. So I again have a video where I can show you and explain to you how the coating is done. So this is the matrigel aliquot. As you can see here, it's kept in ice, as I mentioned earlier. And this matrigel has phenol red, that is why it's appearing pinkish red. Again, as you can see, this is the cell culture hood or cell culture lab. So as we discussed earlier, the laminar air flow has been switched on so that like all your contaminating air will be outside only, it will not go inside. So an important thing is, because your matrigel is going to form start forming a matrix if it goes at a higher temperature, that is if it goes above 10 degrees Celsius, it will start forming a matrix. So important thing is to use a cold medium. So here the DMEM medium that you can see, which is kept here, it should be cold, that is it should be somewhere around 4 degrees Celsius, so that when you add your matrigel here, what will happen? It will not start forming matrix even before it has been plated onto the cells. So these round dishes which you are seeing here, these are the culture dishes which we are going to use for our coating of matrigel. So these are small 2 centimeter dishes, 2 centimeter petri plates. So we have this cold media here. Again, you can see the slurry pipette, and these slurry pipette are one-time use only. And this is so that contamination is prevented. You don't use the same pipette again and again when it is exposed to air. So you can see she has taken the cold media here, and that she will aliquot it out in a falcon tube. So once this is done, so she's pipetting it up and down for proper mixing, and now she'll add the matrigel. You can see it's such a small aliquot. It's somewhere around only 10 to 20 microliters. And she has added that to a 15 ml media. And now she'll invert the falcon up and down for proper mixing of the matrigel with the cold media. Now once this is done, in order to coat the plates, the simplest thing to do to coat the plates will be to pipe this out and put it on the like base of the dishes. So as you can see here, she's pipetting the matrigel plus the medium solution out on the surface of the dishes. And then she's swirling the plates for an even mixing or proper disposal of the matrigel and the medium mixture on the even for the even distribution of this mixture. And once this is done, the plates are again kept at 37 degrees Celsius for somewhere around 1 hour, so that like the matrix is formed. So in this situation, when we have just taken out our matrigel from minus 80 degrees Celsius and our medium is also cold somewhere around 4 degrees Celsius, here the matrix will not be formed. The matrix will be formed only when you keep it at higher temperature, somewhere around 25 degrees Celsius or 37 degrees Celsius. So in the cell culture lab, we have these incubators which have a temperature of 37 degrees Celsius, and that is why we for coating we keep the after adding matrigel to the plates, we keep the cells at we keep the plates for coating at 37 degrees Celsius. So this is how in laboratory matrigel is used for coating. So now moving on to the next question. If I want to conduct an experiment which is mimicking moon or space environment, what should I use? So now an important thing to consider here is mimicking moon or space environment. That means the gravity is going to be zero or the gravity is going to be very less. So, so this is the hint in the question. Now the options are clinostat, weighing balance, thermometer, or a fluorescence assisted cell sorter. So you can write your answers in the chat box. Yeah, so the correct answer is a clinostat. So what is basically a clinostat? So a clinostat is basically a freely rotating device which mimics a zero gravity scenario or microgravity. So Manik sir has discussed this in detail in one of his video lectures. I hope you have watched it. If not, you can go back and watch it again. I'll just explain very briefly how a clinostat works. So what happens, since it's a free rotating device, it will rotate this entire 360 degrees, and like it will not entirely in all its perfection mimic a zero gravity situation, but it there will be some small gravity, and hence technically it mimics microgravity rather than zero gravity. So as you can see here, this clinostat that I am showing you here, this is a 3D clinostat. And you can see this petri dish here. and in the petri dishes we have our seeds. And so basically a clinostat is used in plant cell culture, where we have to know what is the effect of microgravity or no gravity on the growth of plants. So here we have our seeds, like in these petri dishes. These are very small, that is why you cannot, I guess, see them clearly here. But so basically this is a 3D clinostat, and then it will rotate all 360 degree. And now this is a 2D clinostat, where what will happen, your cells will be seeded on cover slip or fixed on the cover slip, and this cover slip can then be added to, like as you can see, these are the rotors or rotating blades which are present, and now this will rotate your entire 360 degree. And once you have inserted the cover slip in the chamber, here you can add your medium. As you can see, this is your medium with phenol red. So this is a very small clinostat, 2D clinostat. As you can see, it just looks like a box. And then you can add it here. See, you can add it, this will be inserted here. And then it will be rotated continuously to simulate microgravity. So clinostat is majorly used in plant cell culture or plant cell biology, but this is an example of a 2D clinostat where it can be used for our normal, like for normal cell culture as well, depending on the kind of application that you want to have and why do you want to study the growth of your cells in microgravity. So next question, MEMs. MEM stands for macro electromechanical system, micro electromechanical system, micro electromagnetic system, or macro electromagnetic system. So I think this was discussed in one of the cell culture lectures. Yeah, so the correct answer is B, that is micro electromechanical system. But in animal cell culture, these days there is something else also which is an MEM, and that is minimal essential medium. So it's just kind of a basic medium which has your like the minimal requirements, amino acids and things like that. And it is also abbreviated MEM. I have just mentioned it because this term is very common in animal cell culture systems. So just in order to avoid any confusion. But here in this context, MEM stands for micro electromechanical system. Now, what is MEM? So, if you would have listened to Sir's lecture, you would have heard something, you would have heard a term, lab on a chip. So, MEM is basically used to create small integrated devices, and these devices can range in size from a few micrometers to millimeters. As you can see here, so this is an example of a chip. So this example is a chip for a technique called FISH, which is fluorescence in situ hybridization. I'll not go into the details of the technique. It's just that, so instead of doing everything in very big culture flask or very big culture vessel, what you can do, you can just have it in something as small. So as you can see here, this device which is used for doing FISH is as small as a chip or as small as a cover slip. And here the small holes that you are seeing, these are very small chambers which, as you can see, are only millimeters long. And here in every chamber is specialized for a different kind of process which you do separately in your lab. So this is what MEMs are a current trend, and this is the common term which is used for that, which is lab on a chip. So it can have different bioMEMs can range in different domains. We can have an organ on a chip, a microfluidic cell culture. Again, microfluidic cell culture will be where there is something like this, very small, and then you have your micro channels which can pass on your microfluids. And then we can have PCR chips where PCR can be done into something as small as a chip. Again, there will be different chambers where different kinds of things can be done, but it will be just one chip where the entire PCR reaction can be carried out. So you can appreciate like MEMs are something kind of a futuristic technology where instead of having big equipments or like using too much of components, we can just perform that reaction or that assay on something as small as a chip. Now the next question, fluorescence occurs by emission of electrical waves only, magnetic waves only, electromagnetic waves, or none of the above. So if you know, you can write the answer in the chat box. Yes, so the correct option is fluorescence occurs by emission of electromagnetic waves. So I think everyone knows this because this is basic class 10 science. But again, I'll just show that an electromagnetic wave has both an electric field and a magnetic field oscillating perpendicular to each other. So fluorescence or any kind of visible radiation that we see is because of electromagnetic wave. Moving on to the next question, osmolarity is total number of solute particles per kg of solution, total number of solute particles per liter, moles per kg, or number of moles. Yes, so the correct option here is B. Osmolarity is total number of solute particles per liter. However, total number of solute particles per kg of solution will be something which is called osmolality. So this is the difference. But here for this question, osmolarity will be the total number of solute particles per liter. Since we are talking about osmolarity, a very basic and important concept to discuss is about different kinds of osmotic solutions. So these are different kinds of osmotic solutions. Sorry. So the first kind of osmotic solution is an isotonic solution. So as the name suggests, iso means something which is balanced. So here the solute concentration inside your cell will be same as the solute concentration outside in the environment. So here your cells will be very happy, they'll be growing normally because the solute concentration is the same, and hence there will be no movement of water in the cell or out of the cell. However, in a hypotonic solution, what happens is like the solute concentration inside your cell uh, will be high, and hence like water will get inside your cell, and in such a case what will happen is your cells will get life. In a hypertonic solution, there will be a very high solute concentration, as a result of which water will get out of the cell and your cell will look shriveled. So these are the different kinds of osmotic solutions. Now coming to osmolarity. So we all know about what molarity is, right? It is the like number of solutes, number of molecules of solutes which are found per liter or per unit volume of your solution. In similar way, osmolarity is the same. So osmolarity does not depend on the kind of solute particles which will be present. So osmolarity will have no relation to the kind of solute particles. You can have the same kind of solute particles. You can have different kinds of solute particles. However, osmolarity will depend on the total concentration or the total number of solute particles which are present. So this is a very important concept. You can have questions like this also, that does osmolarity depend on the uh, two solutions which have an equal number of solute particles, but one solution has X solute particles and the other solution has X plus Y solute particles. So will the osmolarity be different? The answer will be no, because if the number of moles or the number of solute particles is the same, the osmolarity is going to be the same in a specified volume of solution. So moving on to the next question, osmolarity is measured using is osmolarity measured electrochemically? Is it measured using freezing point depression, weighing balance, or immunocytochemistry? So here the correct option will be B, as people have mentioned in the chat box. Osmolarity is measured using freezing point depression. So now here, so this what you are seeing is a basic setup for measurement of osmolarity. So this is something which is called freezing point osmometry. Osmometry, as the name suggests, is just a measurement of your osmolarity. So what happens is, why are we using freezing point depression for measuring osmolarity? So what happens is, the freezing point of your solution is going to decrease as you are going to add solute to your liquid or to your solution. So in the graph, as you can see here, if the freezing point is going to be decreased and if it is going to be cooled, what will happen? Uh, and the freezing point is decreasing because you are adding a particular solute. And so basically, the depression is somewhere around 1.8 Kelvin per mole of solute that you add. So suppose if you know the freezing point of that solution prior to adding your solute, and suppose you add 2 moles of your solute, and then you you will think what will be the osmolarity of that particular solution. So you can measure the freezing point, and depending on that, you can back calculate the osmolarity of your solution, because you can compare the different freezing points, and this is how you can calculate the osmolarity as well. So the next question is, cell attachment can be promoted by treating the substrate with cold trypsin, warm trypsin, polylysin, or an antibiotic. So if you know the answer, you can mention in the chat box. So yeah, the correct option is C. It's not A. Cold trypsin does not aid in cell attachment. Trypsin is a enzyme which is used for detachment of cells. So it's a protease. Trypsin is a protease. So it will not aid in cell attachment whether it is cold or warm. It will aid in cell warm. Trypsin is going to aid in cell detachment. So here, for this question, cell attachment is promoted by treating the substrate with polylysin. So polylysin is a synthetic polymer. So this is the basic diagram of how polylysin looks like, and this N can be the number of units that are going to be repeated. And what this does, this is in cell culture, polylysin is the most commonly used synthetic polymer for aiding in cell attachment. And how does it do that is, so your polylysin is positively charged and your cell surface is going to be negatively charged. Your cell surface or your DNA is going to be negatively charged. So suppose this is,
So
so
this
this
is
is
your
your
substrate.
substrate.
Just 1 second. So basically, this is your substrate, and here it's mitochondria, but here can be our cell also. So the cell membrane is negatively charged, and because of this positive negative interaction, polylysin aids in attachment of the cells to your surface. But keep in mind that polylysin is not an ECM. It's not an ECM. It's just a synthetic compound which helps in attachment because of this charge distribution. So here, as you can see, if you have a glass substrate, your tea culture flask or your petri dish, then it will have different kinds of charges, and since it is positively charged, it will go and attach to them. So this is the polylysin positive charge. And here, if you add your DNA or you add your cells, because of this opposite charge interaction, it will get adhered to the surface coated with polylysin. So this these are the 10 questions for today, and another thing is that you can so we upload all these assignment videos on YouTube, so you can go to the channel, and if you have missed any video or any session, you can directly watch the recording. Still, for your reference, I am adding the link on the chat box, so you can see it from there also. So this is all for the questions today. If you have any additional questions, you can answer. You can put those questions in the chat box. I think we still have 2 minutes. T20 area in centimeter square or millimeter square. So, in standard nomenclature, it is in centimeter square, like T20 20 centimeter square. But of course, you can convert it to millimeter square. But when I'm saying T20, I mean 20 centimeter square surface area. So another thing is that like the presentation which is which I showed you will also be shared with you. So the NPTEL team will be adding that presentation in a Google Drive, and it will be shared with you. Sir discussed in one of the lectures that it is millimeter. It's not millimeter, it's centimeter square. You can check it again, or if you still have confusion, you can maybe post it in the discussion forum, and I can also check it again in the lecture where he has mentioned millimeter. Maybe he has mentioned some dimension in millimeter and not specifically the area. So this is all for today's session. uh, Application of clinostat. So as I mentioned earlier, like It is used for simulating microgravity. Suppose you want to see how a kind of cell or a kind of plant. So it's mostly used in plant cell culture. You want to see, there's something called geotaxis, right? Like what is the behavior of the growth of a plant towards gravity. Now you want to, suppose you want to see that in the absence of gravity, if you're going to provide it everything but just not gravity, how your plant is going to grow. Or if there is something like microgravity, then how your plant is going to grow. So to study those kind of environments, a clinostat is used. That is the application. Since it is a flask, why not volume? Okay, so basically you can talk in terms of volume as well. So if you remember, I'll share the slide again. So here, as you can see, I shared a table where this table where you can see for different kind of tea flask, you can see the different kind of volumes which are required. But in a standard notation, volume is not used. Suppose I am saying I have to use a tea flask which can accommodate up to 5 ml volume, but I am not going to call it T5. And the reason for that is because in even if the maximum even if the volume that it can accommodate is up to 5 ml, still it can happen that for my application I am just going to use 2 ml medium and I'm not going to use 5 ml. So in order to avoid that confusion, we like the surface area, like the surface area is going to be constant, right? Because it's just a flask. But volume, because you have to add that particular volume inside your flask, so volume is going to be different depending on your own experiment or your own assay. That is why you can, you for your personal communication, you can talk in terms of volume, but standard notation is not with volume. So thank you very much for attending this session today.